Hello students, welcome to ePathshala. I am Sabah Khan and I will be teaching you therapeutic response and toxicity. In therapeutic response and toxicity, we will be discussing about introduction, combination effect of drugs. In combination effect of drugs, we will be learning about antagonist, receptor antagonist, other types of antagonist. We will be learning about partial agonist, inverse agonist. We will be discussing about potentiation. Then we will discuss about regulation of receptors, and at last we will be talking about toxicity. So let's start with therapeutic response and toxicity. Most drugs exert their effects by interacting with receptors, that is specialized target macromolecules present on the cell surface or intracellularly. A general property of all receptors is their ability to interact with their endogenous ligands or hormones or neurotransmitters to alter cellular responsiveness without changing the chemical nature of the ligand. Cells may have 10 or thousands of receptors for certain ligand or drugs. Cells may also have different types of receptors, each of which is specific for a particular ligand. These ligands can be drugs also. So, the drug receptor complex has basically three elements. What are these three elements? Receptors as determinants of the quantitative relation between the drug concentration and pharmacological response. Receptors are also regulatory proteins that provides target for the drugs. Receptors are the key determinant of therapeutic and toxic response in a patient. Now we will be discussing about occupancy theory of drug response. What is this occupancy theory of drug response? It is the biological or functional response to receptor activation that can be assumed to be directly proportional to the number of receptors occupied by a given ligand at equilibrium. Now the given equation is used to describe this occupancy theory of drug response, in which EF is the fractional response, EM is the maximal response, RL is the concentration of receptor ligand complex, RT is the total receptor concentration. The response elicited by receptor occupation is directly proportional to the number of receptors occupied by the ligand. The equation given on the screen describes the equilibrium condition and the dissociation constant. Now what is dissociation constant? The equilibrium dissociation constant gives a measure of the affinity of the ligand for the receptor. This is the same equation that is derived from law of mass action. The occupancy theory can be described in the following way. The receptor drug complex is reversible. Association of drug and receptors is a bimolecular process. Dissociation of drug and receptors is a monomolecular process. All receptors behave independently of one another. The concentration of drug is greatly in excess of the receptor number, and therefore the binding of the drug to the receptor does not alter the free concentration of the drug. The therapeutic response of a drug is directly proportional to the number of receptors occupied by the drug. So, the affinity of a drug for the receptor is measured by its dissociation constant instead of association constant. The equation of dissociation constant is already given on the screen. So, when a receptor is occupied by an agonist, the resulting drug receptor complex conformational change is the only first of several steps generally required to produce a pharmacological response. The transduction process between receptor occupancy of a drug to exhibit its pharmacological response is called coupling. The initial conformational change in the receptor by a drug is partially responsible to determine effectiveness of occupancy response coupling. Thus, a full agonist is considered as more efficiently coupled to receptor occupancy than other types of drugs such as partial agonists. So, the coupling efficiency is also determined by the biochemical events that transduce receptor occupancy into cellular response. However, receptor occupancy theory is not appropriate for conditions that involve spare receptor and the receptor reserve. We have talked about the spare receptors. What are these spare receptors? So, the maximal response of most tissue to powerful natural agonist is produced by partial activation of the total receptor density, and the remaining receptors appear to be spare, and in fact they participate in making the tissue more or less sensitive to the agonist. Let's understand this with the help of an example. The ileum of guinea pigs produce maximal force of isometric contraction to histamine by activation of only 3% of the available receptors. Therefore, in practical terms, 97% of the receptors are spare receptors and can be removed or inactivated with no diminution of the maximal response to histamine. Moreover, therapeutic response of drugs is facilitated by either receptor or non-receptor mechanisms. These non-receptor mediated drug responses are purely by physical or chemical means, changing the enzyme transporter activity or ion channels. Some of the examples of non-receptor mediated drug actions are antacids, neutralizing the excessive hydrochloric acid in stomach. So, this was the example of non-receptor mediated drug action. More examples include charcoal inhibiting the intestinal absorption of toxins by absorption, isafgula husk used as laxative due to its water retention property in colon, aspirin inhibits cyclooxygenase enzyme for its analgesic effect. In addition, drugs like purine, pyrimidine analogues like antimetabolites exert anti-cancer immunosuppressant activity, and alkylating agents that covalently binds to nucleic acid and produce cytotoxic effect in cancer cells. These are the examples of non-receptor mediated mechanisms. Now we will be learning about drug receptor interaction. It is very important to learn about how drugs interact with receptors. Before this, we need to learn about the certain terminologies like affinity, intrinsic activity. What are this affinity and intrinsic activity? Affinity. Affinity is the ability of the drug to get bound to the receptor. Now what is intrinsic activity or efficacy? Intrinsic activity is also called efficacy. This is the ability of a drug to elicit a physiological response when it interacts with a receptor. Efficacy is dependent on the number of drug receptor complexes formed and the efficiency of the coupling of receptor activation to cellular responses. Here a term comes agonist. What is this agonist? Agonist is a drug or molecule that bind to physiological receptors and mimic the regulatory effects of the endogenous signaling compounds. In other terms, an agonist has maximal affinity towards receptor and elicits the maximal intrinsic activity. Now we will be learning about the combination effect of drugs. Combination effect of drugs include antagonist, partial agonist, inverse agonist, synergistic and potentiation. What is antagonist? When the two drugs are administered together, one drug, that is antagonist, abolishes or it reduces the action of another drug. That another drug can also be called as agonist. Antagonists have high selectivity towards receptors without any intrinsic activity. The antagonism can be of two types. It can be either receptor antagonism or it can be some other type of antagonism. What is receptor antagonism? The drugs bind to receptors without regulatory effect and block the binding of the endogenous agonist. For example, atropine binds to muscarinic cholinergic receptors in intestinal smooth muscles and inhibits the contraction of muscle. This is the example of receptor antagonism. Receptor antagonism can also be competitive or non-competitive type. Let's understand the concept of competitive antagonism. Antagonists binds reversibly to the same receptor site as the agonist binds. The inhibitory effects of antagonist can be surmounted by addition of a higher concentration of agonist because antagonists bind reversibly to the receptor site. In the presence of competitive antagonist, this produces a rightward parallel shift of the dose response for the agonist, that is towards higher concentration, and agonist can still produce the same maximal effect as in the absence of an antagonist. However, higher agonist concentrations are needed to achieve the maximal response. So, the most of the clinically used drugs that act as receptor antagonists are competitive antagonists. For example, isoprotenolol and propranolol bind reversibly to the same receptor subtype, that is beta adrenoreceptor. D-tubercularine, competitive antagonist, and acetylcholine endogenous ligand bind reversibly to the nicotinic cholinergic receptors. Now, what is non-competitive antagonism? Here, antagonists either bind irreversibly, that is by covalent bonding to the receptor site, or bind to a different site of a receptor, which reduces the affinity of the agonist by an allosteric mechanism. So, what we have learned that in competitive antagonism, antagonists bind reversibly, but in non-competitive antagonism it binds irreversibly. In the presence of non-competitive antagonist, reduction in the maximal response produced by the agonist is observed. In contrast to a competitive antagonist, the effect of a non-competitive antagonist cannot be reversed by simply increasing the agonist concentration, since the law of mass action does not apply here. For example, phenoxybenzamine is a non-competitive antagonist and it binds covalently irreversibly to alpha adrenergic receptors. On increasing the noradrenaline concentration in many folds, the maximal response cannot be reproduced as in the absence of an antagonist. The figure appearing on the screen describes the dose response curve of competitive and non-competitive antagonist. There are other type of antagonisms also, for example, physiological antagonist. The two drugs act on different receptors or by different mechanism but have opposite overt effects on the same physiological function, that is, has pharmacological effect in opposite directions. This is called as physiological antagonism. We have learned about physiological antagonism. Let's understand this with the help of an example. Norepinephrine increases blood pressure via vasoconstriction. It can be antagonized by acetylcholine, which causes vasodilation by stimulating the muscarinic receptor. So, this was the example of physiological antagonism. There are other types of antagonism also. For example, chemical antagonism. What is chemical antagonist? A drug is called as chemical antagonist when it interacts chemically and inhibit or inactivate the other active drugs. For example, chelating agents like calcium disodium EDTA, BAL form complex with several compounds including toxic metals in GI tract. These chelating agents are chemical antagonists to toxic metals. Protamine sulfate is used as an antidote for heparin overdose. Protamine sulfate is a positively charged substance when administered intravenously. It binds to heparin, which is a strong negatively charged substance. So, this was the example of chemical antagonist. There are pharmacokinetic antagonists also. What are these pharmacokinetic antagonists? When a drug alters the absorption, metabolism or elimination of another drug, then it is called as pharmacokinetic antagonist. For example, in alkaloidal poisoning, activated charcoal is used to adsorb the alkaloid present in GI tract and thus reduce their absorption in intestine. Phenobarbital, it is a potent microsomal enzyme. It is an enzyme inducer and thus increases the metabolism of other co-administered drugs. Coumarin, an anticoagulant, it metabolizes faster when co-administered with phenobarbital in patients. There are other types of agonists also like partial agonist, inverse agonist, Now let's understand these terminologies, what are partial agonists and what are inverse agonists. Partial agonists are the agents that are only partly as effective as agonists irrespective of their doses. These drugs have affinity but show submaximal intrinsic activity. For example, pindolol is a non-selective beta blocker with partial beta adrenergic receptor agonistic activity. What are inverse agonists? These drugs stabilize the receptor in its inactive conformation. They produce a response that is opposite of that typically produced by a receptor when it is stimulated by a conventional agonist. So these drugs have affinity but show negative intrinsic activity. For example, agonists for the GABA receptor such as benzodiazepines create a sedative effect, whereas inverse agonists have anxiogenic effect or even convulsive effect. For example, certain beta carbolines. Synergism. What is synergism? Synergism is when the therapeutic response of two different drugs with similar effect is higher than the total response of two individual drugs. Then it is called as synergism. So when two drugs are combined, their therapeutic response is greater as compared to their individual therapeutic responses. For example, combination of paracetamol and codeine as analgesic agent and also amlodipine and atenolol as antihypertensive agent. Similarly, there is another term called potentiation. Potentiation is when one inactive drug augments the therapeutic response of an active drug and the response is increased more than one-fold. For example, levodopa and carbidopa combination for treatment of Parkinson's disease. Carbidopa is an inactive drug, however, it enhances the effect of levodopa by increasing its availability in brain. So this was the example of potentiation, how a drug potentiates another drug's therapeutic activity by the potentiation mechanism. Regulation of receptor is a very important concept to understand. What is regulation of receptors? Now let's understand this. Receptors not only initiate regulation of biochemical events and physiological function, but they also are themselves subject to many regulatory and homeostatic controls. These controls include regulation of receptor synthesis and degradation by multiple mechanisms, association with other regulatory protein, covalent modification, and relocalization within the cells. Effector proteins and transducers are regulated similarly. Modulating signaling inputs may come directly or indirectly from other receptors and or feedback regulation by their own signaling outputs. The variation in different therapeutic response of a drug could be due to the following mechanism involved in receptor regulation. These mechanisms are down regulation of receptors and up regulation of receptors. What is down regulation of receptors? Continued stimulation of cells with agonists generally result in a state of desensitization, such that the effect that follows continued or subsequent exposure to the same concentration of drug is diminished. This phenomenon is called as tachyphylaxis. It occurs rapidly and is therapeutically important. Let's understand this with the help of an example. Repeated use of beta adrenergic receptor agonists such as bronchodilators for the treatment of asthma. The dose of beta adrenergic receptor agonist needs to be increased for getting the same therapeutic response as during the initial period of treatment. Desensitization can result from temporary inaccessibility of the receptor to agonist or from fewer receptors synthesized and available at the cell surface. For example, downregulation of receptor number. Phosphorylation of the receptor by specific GPCR kinase play a key role in triggering rapid desensitization. What is upregulation of receptors? In downregulation, we have learned about desensitization. Now what will happen in upregulation? It is supersensitization. So, supersensitivity to agonists also frequently follow chronic reduction of receptor stimulation. Supersensitivity is mainly due to the upregulation of receptors at the cell surface. Such situations can result following the withdrawal of an antagonist from prolonged receptor blockade. For example, the long-term administration of beta-adrenergic receptor antagonists such as propranolol. Abrupt cessation of propranolol therapy can lead to sudden increase in the heart rate and blood pressure. Supersensitivity also can be the result of tissue response to pathological condition such as it happens in cardiac ischemia and is due to synthesis and recruitment of new receptors to the surface of myocyte. Toxicity. What is toxicity? Toxicity is defined as any toxic or adverse effect that a chemical or physical agent might produce within a living organism. Research scientists essentially stated this concept as all substances are poisons, there is none which is not a poison. The right dose differentiates a poison and a remedy. Drug toxicity usually occurs at doses that exceed the therapeutic efficacy of a drug. For example, coma can occur by barbiturates overdose, complete AV block can occur by digoxin overdose, bleeding due to heparin, morphine causes respiratory failure in overdosage. So, the dose-response relationship provides the basis for estimating the safe exposure level for a chemical. Dosage of drugs are often described as lethal dose, toxic dose and sentinel dose. What is lethal dose, what is toxic dose and what is sentinel dose? Lethal dose is where the response being measured is mortality. In toxic dose, where the response is a serious adverse effect other than lethality. Sentinel dose is when the response being measured in a non or minimally adverse effect. Sentinel effect examples are minor irritation, headaches and drowsiness. Dose response curve of a drug with different dosage forms are the sentinel response curve. It might represent a relatively safe acute toxicity such as odor or minor irritation to the eyes or nose. The toxic response or TD curve, it might represent a serious toxicity such as organ injury or coma. Then there is a lethal response curve. Lethal response or LD curve represents doses that produce death. Margin of safety can be defined as TD50 upon SD50, that is toxic dose and sentinel dose at their 50% concentration. There is a term called margin of safety. What is margin of safety? Margin of safety represents the difference in dose between the toxicity curve and a sentinel effect curve. The margin of safety is calculated by dividing TD by the SD. The higher the margin of safety, safer the chemical to use. Following figure on the screen describes the comparison of several dose response curve for a toxicant. So, the drug toxicity refers to the level of damage that a drug can cause to an organism. The toxic effects of a drug are dose dependent and can affect an entire body system as in the CNS or a specific organ such as the liver. Toxicity effect of a drug is usually observed when the dose exceeds the therapeutic efficacy. There are several examples of toxicity that are extension of therapeutic effect itself. For example, barbiturates are the CNS depressant which leads to coma. Heparin is an anticoagulant which has common toxicities like hemorrhage, bleeding. These are the example of toxicity that are linked to other action of the drug but not related to their therapeutic effects. For example, overuse of imipramine as antidepressant can cause cardiac arrhythmia and prolonged use of streptomycin as antibiotic can cause ototoxicity. Next, we will be discussing about manifestation of toxicity. For example, phototoxicity. This is also a manifestation of toxicity. In phototoxicity, skin response to light is altered due to deposition of certain drugs and their metabolite in skin. The accumulated drug in skin aggravates photosensitivity due to photochemical reaction followed by their photobiological reaction. Sun-exposed skin area of the body is more sensitive and sunlight with UVB wavelength, that is 290 to 320 nanometer, are responsible for these type of reactions. The consequences of phototoxicity are local tissue damage, that is irritation, blistering, erythema, edema that leads to hyperpigmentation and desquamation. Drugs that can cause phototoxic reactions include tetracycline, ibuprofen, ketoprofen, furosemide, nalidixic acid, fluoroquinolones, sulfones and sulfonamides. Next is pulmonary toxicity. Drugs in the form of gases, solid particles or liquid aerosols are exposed throughout the respiratory tract and may damage the tract depending on their physical and chemical properties. Drugs may cause number of respiratory diseases upon exposure to lungs that include asthma, bronchitis, emphysema, hypersensitivity, pneumonitis, pneumoconiosis and cancer. After the damage induced by drugs, lung alveolar epithelium are repaired and replaced by fibrous tissues. The alveolar ventilation of lungs decline tremendously after the fibrous tissue deposition. Next is hepatotoxicity. Most of the orally administered drugs or toxicants are absorbed from stomach or small intestine that are transported directly to the liver through portal vein. Thus, liver is exposed with relatively large quantities of ingested drugs or toxicants. Drugs such as paracetamol or chloroform undergoes biotransformation to toxic metabolites within liver. Those toxic metabolites necrotize the cells surrounding the central veins where cytochrome P450 enzyme system are found abundantly. Acute exposure with hepatotoxin with a single large dose may cause hepatic necrosis. However, the damage is resolved with minimal or no tissue scarring, whereas chronic exposure can result in liver cirrhosis and permanent tissue scarring. The liver damage vary upon the types of drugs exposed to the body. For example, estrogen can cause mild cholestatic liver damage, whereas chlorpromazine can cause severe cholestatic liver damage including inflammation and hepatocellular necrosis. Next is nephrotoxicity. Most of the drugs and their metabolites are excreted through the kidney, which have high renal blood flow. Thus, kidney is the most susceptible organ to xenobiotic toxicity. Upon the exposure of drugs and their metabolites, the extent of damage may vary in different parts of nephron. Tubular cells are more vulnerable to damage due to their double-sided exposure to drugs or metabolites, whereas basolateral side is exposed with toxin present in blood and luminal side is exposed with toxins present in urine. Further, proximal tubular cells have abundance of cytochrome P450 enzyme and organic anions cation transporters. Thus, toxins are concentrated many folds that cause maximum damage to tubular cells, which is typically seen as acute tubular necrosis. Impaired absorption of water, electrolyte and essential nutrients due to tubular cell damage results in compromised feedback mechanism and thus reduction in glomerular filtration and accumulation of large volumes of water. Urine output is not a predictive indicator for analyzing the extent of renal damage. Thus, biological marker like creatinine and blood urea nitrogen is used to predict the changes of glomerular filtration. Next is neurotoxicity. Unlike the other vital organs, brain and nerves are well protected from exogenous substances. Central nervous system and peripheral nervous system is protected by the blood-brain barrier and blood-neural barrier respectively. However, these barriers are ineffective against highly lipophilic compounds such as solvents and insecticides. Further, brain and neurons are considered to be susceptible for neurotoxin challenge because of their high perfusion rate, metabolic rate and lipid content. In case of fetus and neonates, the nervous system and their barriers are not well developed that cause easy passage of neurotoxins to brain and neurons. Regeneration of neural tissue after injury is difficult. Subsequently, other non-conducting cells and glial cells proliferate and occupy the area of dead neurons. Neuronal damage may be noticed as deficits in motor and sensory function as well as changes in behavior. Alternatively, other healthy neurons may undertake the function of damaged neurons, such as the neuronal damage is minimal or non-evident. Next type of toxicity is immunotoxicity. In this, immune system is impaired upon exposure to number of drugs and chemicals that are environmentally and occupationally important. One or more component of the immune system is affected by these xenobiotics that result immunodeficiency or allergic autoimmune reactions. Clinical signs and symptoms of cutaneous allergic reactions are itching, urticaria, eczema, dermatitis and skin eruption. Sometimes drug allergic can affect body system causing breathing problem, wheezing and anaphylaxis. Allergic reactions involving immune system is different than the allergic reactions caused by direct irritant. An important difference is that initial exposure to a drug chemical can cause sensitization of the individual's immune system. Second time, further exposure to a minimal quantity of the drug or chemical elicits the allergic reaction. For example, the drugs that cause drug allergy are amoxicillin, penicillin, aspirin, insulin, etc. Other forms of immunotoxicity is immunodeficiency. Immunodeficiency is the loss of immune response to infection, precancerous or cancerous cells and other tissue damaging factors. Therefore, the organ toxicity of selected drugs are shown on the screen. A very important manifestation of toxicity include teratogenicity. Most drugs and chemicals pose a threat to the developing fetus. An estimated 4 to 5% of developmental defects in human results from prenatal exposure to drugs or environmental chemicals. Drugs can affect the fetus in 3 stages. First is fertilization and implantation. Conception to 17 days, failure of pregnancy which often goes unnoticed. Second stage include organogenesis, which is from 18 to 55 days of gestation. It is the most vulnerable period when deformities are produced. Third is growth and development, that is 56 days onward. Developmental and functional abnormalities occur at this stage. Example of teratogenic incident include thalidomide causes phocomelia, ACE inhibitor can cause hypoplasia of organs especially lungs and kidneys, then NSAIDs may produce premature closure of ductus arteriosus. So, these are the common example of teratogenic drugs. Next we will discuss about the mutagenicity. Mutagenicity is the capacity of drug to cause genetic defects. Xenobiotics are often bioactivated to metabolites that are sufficiently reactive to DNA and then they disrupt its coding. Now what happens in carcinogenicity? It is when capacity of a drug to cause cancer. If the xenobiotic metabolite can modify DNA sequence code for factors that regulate cell proliferation growth, that is proto-oncogenes, or for proteins that inhibit transcription of proto-oncogenes, a tumor may be produced. The examples of carcinogenic drugs are anti-cancer drugs, radioisotopes, estrogen, tobacco, etc. To conclude, in this module, what we have learned? So, we have learned about the drug receptor relation, we have learned about agonist, antagonist, partial agonist, inverse agonist. We have learnt about the different mechanism of antagonism, that is reversible, irreversible, or competitive or non-competitive. And what we have learnt is that if the drug is administered in its overdose, then it can cause serious toxic effect.